clownfish or anemonefish are fishes from the subfamily Amphiprioninae in the family Pomacentridae. 30 species are recognized, 1 in the genus Premnus, while the remaining are in the genus Amphiprion. In the wild, they all form symbiotic mutualisms with sea anemones. Depending on species, anemonefish are overall yellow, orange, or a reddish or blackish color, and many show white bars or patches. The largest can reach a length of 18 centimeters, while the smallest barely achieve 10 centimeters. Distribution and habitat. Anemonefish are native to warmer waters of the Indian and Pacific Oceans, including the Great Barrier Reef and the Red Sea. While most species have restricted distributions, others are widespread. Anemonefish live at the bottom of shallow seas in sheltered reefs or in shallow lagoons. There are no anemonefish in the Atlantic. Diet. Anemonefish are omnivorous and can feed on undigested food from their host anemones, and the fecal matter from the anemonefish provides nutrients to the sea anemone. Anemonefish primarily feed on small zooplankton from the water column, such as copepods and tunicate larvae, with a small portion of their diet coming from algae, with the exception of Amphiprion peridorion, which primarily feeds on algae. They may also consume the tentacles of their host anemone. Symbiosis and mutualism. Anemonefish and sea anemones have a symbiotic, mutualistic relationship, each providing a number of benefits to the other. The individual species are generally highly host-specific, and especially the genera Heteractis and Stichodactyla, and the species Intacmia quadricolor are frequent anemonefish partners. The sea anemone protects the anemonefish from predators, as well as providing food through the scraps left from the anemone's meals and occasional dead anemone tentacles. In return, the anemonefish defends the anemone from its predators and parasites. The anemone also picks up nutrients from the anemonefish's excrement, and functions as a safe nest site. The nitrogen excreted from anemonefish increases the amount of algae incorporated into the tissue of their hosts, which aids the anemone in tissue growth and regeneration. It has been theorized that the anemonefish use their bright coloring to lure small fish to the anemone, and that the activity of the anemonefish results in greater water circulation around the sea anemone. Studies on anemonefish have found that anemonefish alter the flow of water around sea anemone tentacles by certain behaviors and movements such as wedging and switching. Aeration of the host anemone tentacles allows for benefits to the metabolism of both partners, mainly by increasing anemone body size and both anemonefish and anemone respiration. There are several theories about how they can survive the sea anemone poison. The mucus coating of the fish may be based on sugars rather than proteins. This would mean that anemones fail to recognize the fish as a potential food source and do not fire their nematocysts, or sting organelles. The coevolution of certain species of anemonefish with specific anemone host species and may have acquired an immunity to the nematocysts and toxins of their host anemone. Experimentation has shown that Amphiprion percula may develop resistance to the toxin from Heteractis magnifica, but it is not totally protected, since it was shown experimentally to die when its skin, devoid of mucus, was exposed to the nematocysts of its host. Anemonefish are the best-known example of fish that are able to live among the venomous sea anemone tentacles, but there are several others, including juvenile three-spot jacilus, certain cardinalfish, buckitches goby, and juvenile painted greenling. Reproduction. In a group of anemonefish, there is a strict dominance hierarchy. The largest and most aggressive female is found at the top. Only 2 anemonefish, a male and a female, in a group reproduce through external fertilization. Anemonefish are sequential hermaphrodites, meaning that they develop into males first, and when they mature, they become females. If the female anemonefish is removed from the group, such as by death, one of the largest and most dominant males will become a female. The remaining males will move up a rank in the hierarchy. Anemonefish lay eggs on any flat surface close to their host anemones. In the wild, anemonefish spawn around the time of the full moon. Depending on the species, anemonefish can lay hundreds or thousands of eggs. The male parent guards the eggs until they hatch about 6 to 10 days later, typically 2 hours after dusk. Parental investment. Most anemonefish are protandrous hermaphrodites, meaning they alternate between the male and female sexes at some point in their lives. Anemonefish colonies usually consist of the reproductive male and female and a few juveniles, who help tend the colony. Although multiple males cohabit an environment with a single female, polygamy does not occur and only the adult pair exhibit reproductive behavior. However, if the largest female dies, the social hierarchy shifts, with the breeding male exhibiting protandrous sex reversal to become the breeding female. The largest juvenile will then become the new breeding male after a period of rapid growth. The existence of protandry in anemonefish may rest on the case that non-breeders modulate their phenotype in a way that causes breeders to tolerate them. This strategy prevents conflict by reducing competition between the males for one female. For example, by purposefully modifying their growth rate to remain small and submissive, the juveniles in a colony present no threat to the fitness of the adult male, thereby protecting themselves from being evicted by the dominant fish. The reproductive cycle of anemonefish is often correlated with the lunar cycle. Rates of spawning for anemonefish peak at approximately the first and third quarters of the moon. The timing of this spawn means that the eggs will hatch around the full moon or new moon periods. One explanation for this lunar clock is that spring tides produce the highest tides during full or new moons. Nocturnal hatching during high tide may reduce predation by allowing for a greater capacity for escape, namely, the stronger currents and greater water volume during high tide protects the hatchlings by effectively sweeping them to safety. Before spawning, anemonefish exhibit increased rates of anemone and substrate biting, which help prepare and clean the nest for the spawn. In terms of parental care, male anemonefish are often the caretakers of eggs. Before making the clutch, the parents often clear an oval-sized clutch varying in diameter for the spawn. Fecundity, or reproductive rate, of the females usually ranges from 600 to 1500 eggs depending on the size of the female. Male anemonefish care for their eggs by fanning and guarding them for 6 to 10 days until they hatch. Studies have shown that, in general, eggs develop more rapidly in a clutch when males fanned properly, and that fanning represents a crucial mechanism of successfully developing eggs. This suggests that males have the ability to control the success of hatching an egg clutch by investing different amounts of time and energy towards the eggs. For example, a male could choose to fan less in times of scarcity or fan more in times of abundance. Furthermore, males display increased alertness when guarding more valuable broods, or eggs in which paternity was guaranteed. Females, on the other hand, display generally less preference for parental behavior than males. All these suggest that males have increased parental investment towards the eggs compared to females. In the aquarium, anemonefish make up 43% of the global marine ornamental trade, and 25% of the global trade comes from fish bred in captivity, while the majority are captured from the wild, accounting for decreased densities in exploited areas. Public aquaria and captive breeding programs are essential to sustain the trade as marine ornamentals, and has recently become economically feasible. It is one of a handful of marine ornamentals whose complete life cycle has been closed in captivity. Members of some anemonefish species, such as the maroon clownfish, become aggressive in captivity. Others, like the false percular clownfish, can be kept successfully with other individuals of the same species. When a sea anemone is not available in an aquarium, the anemonefish may settle in some varieties of soft corals, or large polyp stony corals. Once an anemone or coral has been adopted, the anemonefish will defend it. As there is less pressure to forage for food in an aquarium, it is common for anemonefish to remain within 2 to 4 inches of their host for their entire lifetime. Anemonefish, however, are not obligately tied to hosts, and can survive alone in captivity. Taxonomy. Historically, anemonefish have been identified by morphological features, color pattern in the field, while in a laboratory other features such as scalation of the head, tooth shape and body proportions. These features have been used to group species into 6 complexes: clownfish, tomato, skunk, clarkie, saddleback and maroon. As can be seen from the gallery, each of the fish in these complexes have a similar appearance. Genetic analysis has shown that these complexes are not monophyletic groups, particularly the 11 species in the clarkie group, where only A. clarkie and A. tricentis are in the same clade, with 6 species: A. allardi, A. bisinctis, A. chagosensis, A. crosgaster, A. fusca A. latifasciatus and A. omonensis being in an Indian clade, Chrysopteris having monospecific lineage, and A. echindinose in the Australian clade with A. mccullochii. Other significant differences are that A. latizonatus also has monospecific lineage, and A. nigripes is in the Indian clade rather than with Achylopesos, the skunk anemonefish. A. latizonatus is more closely related to A. percula and Premnus B. aculeatus that to the saddleback fish it was previously grouped with. It is thought that obligate mutualism was the key innovation that allowed anemonefish to radiate rapidly, with rapid and convergent morphological changes that were correlated with the ecological niches offered by the host anemones. The complexity of mitochondrial DNA structure shown by genetic analysis of the Australian clade suggested evolutionary connectivity among samples of A. echindinose and A. mccullochii that the authors theorize was the result of historical hybridization and introgression in the evolutionary past. There were 2 evolutionary groups with individuals of both species detected in both, thus the species lacked reciprocal monophyly. There were no shared haplotypes between species. In popular culture, in Disney Pixar's 2003 film Finding Nemo, the main character is a clownfish. The species depicted in the film is A. ocellaris. It will also appear in the upcoming sequel Finding Dory. Gallery. Clownfish A. ocellaris. A. percula in a normal, orange and a melanistic blackish variant. Clarkie complex A. clarkie. A. echindinose. A. allardi. A. bisinctis. A. chagosensis. A. chrysogaster. A. chrysopteris palau. A. fusca A. latifasciatus. A. omonensis. Tomato complex A. Amphiprium. A. barbari. A. frenatus. A. mccullochii. A. melanopus. A. rubrocinctis. Saddleback complex A. latizonatus. A. Polymnus of Sulawesi, Indonesia. A. sebe. Skunk complex A. Achylopesos. A. leucochranos. A. nigripes. A. peridorion. A. sandarachinos. Premnus B. aculeatus P. B. aculeatus.